So now that we got through the cell's basic anatomy, we can get into a little bit more detail with DNA. So we're going to talk about gene expression really quickly in this video, just kind of go over it briefly. Again, most of this should be a bit of a review, again, we're going to get a little bit deeper into some of this stuff, talk maybe a little bit more about some things that you may or may not have gone over in a previous class. But the general concepts you should feel familiar with. So let's go through it. So we're going to talk about how nuclear DNA is organized, define some terms related to that, talk about the mechanisms of gene transcription, some more terms that go with transcription, and some main players that go with that. And then we'll move into translation, talk about the main players and vocabulary that goes with translation. Then talk about the genetic code and its redundancy, and how we make proteins, and basically how proteins get degraded in the cell and why. So let's start. Let's go with nuclear DNA and start there. So basically, again, like I mentioned in the last video, DNA is stored in the nucleus in the form of what we call chromatin. And chromatin is DNA with its organizing proteins. These organizing proteins are called histones. So we look at the picture here, right? As we zoom in, we see we see the blue strand of DNA, and it's wound around these gray balls called histones. This is going to help us with the organization of DNA so we can compact it together. It's also going to help serve as a regulatory function, basically. It's going to help regulate which genes are expressed and when. We're not going to get heavy in the details of that, but something to keep in mind, that how the DNA is packed together and how it's wound around various proteins is going to affect which genes are used when and how often. So when we do this packing of the DNA together, we do form what we call chromosomes. Chromosomes are going to be like the segments of DNA. If we look at a human, we're looking at 46 chromosomes. And on these chromosomes, we find specific sections of DNA that code for a protein, and these are called genes. Right, so genes are going to code for a protein. Again, we're going to simplify some of this down. This does get complicated, like as a topic. So keep in mind we are going to kind of water it down a little bit. I know that might not feel like that, but we are. And we will be making some assumptions and generalizations that may not hold true all the time. So just keep that in mind as we're going through this. If you read up using other resources and you're like, what the heck, I heard that genes can be used to make more than one protein. And I would tell you, you are correct, but for the sake of simplicity for this class, we are going to keep it as simple as we can without losing too much in the process. So we have these histones that are used to wrap DNA around to form chromatin, and then the packing of that DNA unit around a histone we call a nucleosome. So altogether, the DNA wrapped around the histone is a nucleosome. Now, why do these wrap together? Because histones are charged positively and DNA has a negative charge, and so opposites attract, which causes the DNA to spool around it. Now, some of these sections of chromatin are called euchromatin, which means they're transcriptionally active. Basically, it means that we are actively using those sections of DNA to code for proteins. And so we're going to have a looser spooling right around there, and that's going to allow access to the genes that are found on that section of DNA. And we achieve this through acetylation. So these acetyl groups are added that allow for the the DNA to kind of loosely go around those histones instead of tightly. And then heterochromatin is going to be transcriptionally inactive. Basically, they're super condensed. We're not using those genes at all, if not very often. And so we can afford to pack them away for now, right? We don't need them at that moment. So we look at the picture, right? We see the heterochromatin, it's tightly packed together. So you can see how it'd be really hard to access those genes because like they're really tightly packed. Versus the euchromatin that's active, we see it's much looser, much more open, and so you can see how we'd have an easier time getting access to a particular gene in that more open space in the euchromatin. Now, altogether, the DNA, when we look at the entire set, is called the genome. We're looking at all the genes. In this case, we're thinking about 40,000 in a single individual. This includes the nuclear and mitochondrial DNA. And when we're looking at what we call somatic cells, body cells, so the cells that make up your skin, the cells that make up your muscle and your bones and everything else, right, are all going to have the exact same genetic code, right? Same code. Same code. So you think about that, how we have to have modifications occur in terms of which sections are turned on and off that lead to the different functions of different types of cells. Now, we also have the proteome. The proteome is going to be all the proteins that are made. So it's like the genome, but the protein version. And different tissue types will have different proteomes because some of those genes in, let's say, a muscle cell will be turned on, and if we look at something like a blood cell, those genes are off, right? Basically, we've inactivated the ones in the white blood cells and left them active in the muscle cell. So the proteome, we're looking at what proteins does that cell make based on which genes are turned on, right? So different tissues will have different proteomes, even though we're looking at the same human body, because those different tissues have different functions. We can also see changes in the proteomes under different disease states as well. So if we look at how many genes we have, about 40,000, but we're going to make well over that, right? We're going to multiply that several times over in terms of how many proteins we can make. So again, you can probably do the math. There's not one single gene for every protein. There's things that we can do to modify the product that we get using DNA so that we get more than one protein from that, but we're, again, not going to get caught up in the details. So let's go ahead and talk about gene expression. What does that mean? Gene expression is just looking at making a certain protein using the genetic information we find in DNA. And there's two basic steps: transcription and translation. Transcription, as the name implies, if you transcribe something, right, you write it down in the same, like basically the same language, right? So we're going from a nucleic acid, DNA, and we're using that to form another nucleic acid, in this case mRNA. So nucleic acid to nucleic acid, DNA to RNA, transcription. When we go to the next step, translation, think what translation means, right? If you were to translate what I'm saying right now, you would be translating it into a second language, right? Maybe into Spanish or French or German, right? You would be taking what I'm saying in English and converting it into another language. So we're doing something similar here too, is that we're starting with a nucleic acid, in this case mRNA, and we're translating it into a protein, right? A completely different class of macromolecules. And so we think about what we call the central dogma of biology is that we go from DNA to mRNA to protein, and we don't necessarily go backwards. We have a one-way road with this, and that to get from the DNA to mRNA, we use transcription, and to get from mRNA to protein, we use translation. So this is just kind of demonstrating, just letting you visualize what we're doing, how we're getting there, without any sort of nuance. So let's go ahead and give ourselves some nuance. Let's talk about it. Let's start from the beginning, which is transcription, where we are going to take a gene in the nucleus. We're going to process that gene so that we can create basically our own copy of that genetic information, but in the form of mRNA. And the enzyme that is going to do this action of reading the gene and converting that information into mRNA is called RNA polymerase. So RNA polymerase, we think about the name RNA, we know we're doing something with RNA. Poly or polymer, right? We think like a long chain of many things. A is enzyme. So RNA polymerase is the enzyme that's going to help us build the RNA chain. The gene is the basic unit of heredity. Basically, we have a start codon and a stop codon. Start's going to be at the beginning, stop at the end of the coding region, and that's going to help us, right, produce a protein. A start and a stop is not all we need though. We need other components so we can actually recruit RNA polymerase and get the process going in a timely manner. That's where we have the promoter region. This is going to be before the gene starts, and it's going to basically bind with what we call transcription factors. The transcription factors can then recruit RNA polymerase so we can actually lock on and read that gene. So we look at the steps of transcription. We have 3 main steps, and we'll see these steps listed again in translation, so keep that in mind. They use the same names. We have initiation, elongation, and termination. Initiation, as the name implies, is going to start us off. The promoter binds with the transcription factors, which then allows RNA polymerase to bind. We can start reading our DNA strand and creating our RNA strand. Elongation, RNA polymerase is going to unwind the DNA so that we can have access to one strand of DNA. That one strand becomes the template. That template will then be read by RNA polymerase, and RNA polymerase will bring the complementary bases, the complementary nucleotides, right, in order based on what the coding strand of DNA says. So, for example, if the coding DNA strand says C, then the RNA polymerase will bring G because G is complementary to C. And we'll continue that all the way down the gene, all the way down the line. Once we get to a termination codon, there's 3 of them, UAA, UAG, and UGA, then that's going to tell RNA polymerase to stop, right? You're at the end of the gene, there's no reason to keep going, and so enzymes will then come to stop the process and release the mRNA transcript that we just wrote. Now that being said, we're not done yet because we have this kind of draft, right? We have this what we call pre-mRNA or precursor mRNA, messenger RNA. This is basically the raw information from the gene using transcription, but it's not quite ready. We have some components that don't actually help code for that protein. These are called introns. We need to cut those out. So we need to cut the introns and get rid of them. And then the exons are what's left over. So the exons are the important pieces. These are the parts that actually code for something. And so these are the ones that will get stitched back together when we remove the introns. And then we can go ahead and send this out to go produce a protein. Now, sometimes, remember we have more proteins than we have genes. We can choose which exons to keep depending on what product we want at the end, right? So we do have what we call alternative splicing, which basically is like, hey, like if you look at the picture here, we have exon 1, exon 2, and exon 3. We can choose to just splice together exon 1 and 3 and get rid of 2. And so we get a different protein in that case. Or we keep only 1 and 2, or we keep all 3, or we only keep 2 and 3. All of those differences in how we can splice the exons together can give us multiple end products for the same gene. Then we have to actually make the protein, right? Now we get to translation where we're producing the protein, and so we're going to use the mRNA sequence as a template. We're going to read it and then form a peptide using the complementary base pair rules and the ribosome as kind of the housing unit for this. The whole point with this is to basically read the mRNA to know which amino acids come in and then provide a space where we can form what's called a peptide bond. Basically bond the carbon and nitrogen of amino acids together so we can form this long chain. Now the genetic code is basically our rules of how we read this, right? So how is mRNA read? mRNA molecules are read in sets of 3. So what I mean by that is 3 nucleotides at a time are read, or 3 bases. So think of it like if the English language had words that only were made of 3 letters each, right? You can have lots of different words, but you're limited to only 3 letters per word. It's kind of what we're doing here. Every 3 letters will tell us which amino acid we need next. So we have like about 64 different possible combinations of base letters, right? In their sets of 3. We only have 20 amino acids though. So that means there's redundancy, because if there's 64 possible combinations of nucleotides, but only 20 amino acids, some of those codons, what we call codons, which are the groups of 3 nucleotides, some of those codons are going to end up coding for the same amino acid. Right? Well, this is redundancy, and redundancy is not a bad thing, because that means if something were to change or mutate, it has to mutate in a specific way in order to become a problem. There are cases where we can have a mutation that end up not changing anything at all. So if we look here, right, we have this table. Now let's go ahead and read this table. I'm just going to pick one. Let's say GUC. So first letter G, I'm going to go to G. Second letter is U. So I'm in this row. U is this column, so I'm going to be in this box. GUC, valine. Right, that's what I'm getting out of that. But if you notice, GUU, GUA, and GUG also give us the same amino acid of valine. So because of that, that overlap, we call this redundancy. But it means that if you were to somehow get a mutation in this last letter, no harm, no foul. Nothing changed. You still have the same amino acid in the end, which means you get the same protein too. Now, how do we actually do the building? We have some components. We have the ribosome and we have tRNA that are going to be kind of helping each other out in reading the mRNA strand. So the ribosome is going to bind around the mRNA, but tRNA is going to be the one reading and bringing the correct amino acid. The codon on the mRNA molecule needs to be complementary to what is found on the tRNA. So if the codon, like in this picture, is GCC, then the tRNA has to have CGG, because that's complementary. We call that complementary section on the tRNA the anticodon, because it goes opposite, right, of the codon. So same 3 types of nucleotides, right, but they're just complementary to the codon. And at the very end of our tRNA molecule, we have the amino acid that we're trying to bring in. So when we look at translation, mRNA is going to attach to the ribosome. We're going to read every 3 bases in the triplicate sequence of mRNA. We call that the codon. Each codon tells us what amino acid we need. The tRNA is going to be bringing the correct amino acid based on the anticodon on the tRNA binding with the codon on the mRNA. So what are we doing here? We have decoding. Decoding is where we are basically bringing tRNA to attach to the complementary codon. So anticodon and codon come together. We then have peptide bond formation, where we are going to basically link the amino acid that we just brought to the amino acid that's already there. And then translocation, basically the empty tRNA that's already brought the amino acid by is going to leave, and everything can shift over so we can read the next codon. And we just go through this process over and over and over again, where we are just reading, reading the codon, the right tRNA with the complementary anticodon comes in, it binds its amino acid to the polypeptide chain, and once it's empty, it gets booted out and everything moves down one again. Now, a lot of these proteins are destined to make their way through the rough ER, right? So we can have this happen if we are using a ribosome that's embedded in the rough ER, right? The first 30 amino acids or the leader sequence is hydrophobic, and so it's going to get inserted into that membrane. Then we're going to basically keep pumping out, right, more of that protein, pushing it into the inner layer of the rough ER, the lumen of that. That leader sequence, the first 30 amino acids that get removed, they're basically just there so that we can get started on that rough ER. And then from there, we've built the entire polypeptide. We can transfer it to the Golgi it keeps getting modified if it needs to. So we can add things like a glycan. So we create a glycoprotein, and maybe then we can ship that, and it's gonna be shipped to the outside of the membrane of the cell to help do something with the extracellular space. Now, like we mentioned before, proteins don't need to hang around forever necessarily. A lot of proteins, like regulatory proteins, are rapidly degraded, which means their effects are short-lived, but we want short-lived effects because that means we have better control over the cell's function. That means if we don't need a certain activity to happen anymore, the short-lived nature of the protein means that we can stop that process very quickly. If it had a long life, then we would have a lot harder time stopping certain processes. So think of it like a very sensitive brake. You can stop very quickly when your brake is sensitive, but if you really have to, like if your car is going to take forever to slow down, you're going to have to give yourself way more stopping space or you're going to crash. So we have lysosomal proteases. Basically, the lysosomes can help break proteins down, or the proteasome that we talked about in the last video using ubiquitin can also basically chew up a protein into amino acids that can be used again.